Good afternoon, everyone. My name is Laura Nilsson, and I'm the Associate Dean for Graduate Education in the Faculty of Science here at McGill. On behalf of the Dean and the entire faculty, I would like to welcome you to the third Sun and Science session, which is the sunny summer version of our Soup and Science series. We have three incredible speakers who will be presenting to you today about their research. We have Peter Grütter from the Department of Physics, Professor Tomislav Friščić from the Department of Chemistry, and Virginie Millien from the Redpath Museum. Once the presentations are completed, you will each receive a message from the host to join a breakout room. And in this breakout room, you will be in smaller groups where you will have the opportunity to ask questions and talk with each presenter. Each presenter will rotate through the rooms until they've been in all three rooms, so you'll have a chance to talk to each one of them. After this, we will then head back to the main room for final remarks. So let's get started. We'll begin with Professor Grütter's presentation. Take it away, Peter.
Okay, thanks for the introduction. Let's see, I can't change this. Okay, and welcome to Sun and Science. So my research group builds tools and develops methods. These tools allow you to kind of discover new things, and there's nothing more exhilarating in science than actually making a discovery. Home late at night, early in the morning, and knowing you're the only person in the world that knows about this before you tell your supervisor or your friend or somebody else. And so that has kept my research going on for the last 30 years, essentially. And the tools that we built are the following. It's called an atomic force microscope. The way you measure forces is you have a little spring, and so our springs are little cantilevers, microfabricated cantilevers made out of silicon. They're a micrometer thick, 10 micrometers wide. They have integrated really sharp tips. The radius of this tip is a few nanometers. That's about 5 to 10 atoms across. And when there's a force acting between this tip and a sample, it changes the mechanical status. If you now raster the sample relative to this tip and track the forces, you can generate images. Of course, to do that, you have to integrate your tip here on your spring. Bottom left, this microscope might be in protected environments. That's what you see in the middle picture here. Sarah Burke, a student of mine, looking at a microscope in ultra-high vacuum. Or this is, to the right, this is a microscope that goes down to 20 millikelvins and 18 Tesla fields. You generate an image, and the type of images you can generate are really amazing. Visually impaired, the way you would read Braille. This AFM, this atomic force microscope, measures the atomic forces between a tip and this molecule, pentacene. You can see the 5 carbon rings making up this molecule. You can see the hydrocarbon chain sticking out from it. So you can actually do really impressive structural determination with this atomic force microscope. You can feel the force. Imaged a piece of salt you might have had for lunch, putting on your lunch. You can see the individual sodium atoms. You can see one single atomic step here. You can see that the step atoms, or the force above the step atoms, is a larger. And that's why, stepped surfaces or kinked surfaces here are kind of interesting, because their force field is different, their interaction potentials, their chemical reactivities, catalysis, all those kind of things depend on that kind of structure. So I'm not only interested in actually measuring structure. What we've spent the last 10, 15 years with is actually developing methods, new tools to actually measure properties. So left image with these rings, it actually allows to measure the force between a single electron in the sample and your tip. And so we can count the number of electrons in this particular quantum dot. At the top 3 rings, concentric rings, tells you there's 1, 2, 3 electrons in the dot. The bottom part of it is like a molecule. There's 3 quantum dots here. They're coupled. You can extract coupling strengths, like interaction energies. You can see that this guy has 2 electrons in it. This guy has 1 electron in it. This guy has 2 electrons. So you can actually really do electron spectroscopy with this technique. The right picture, the bottom part is a topography image of a 1 layer thin piece of graphite called graphene. And this was measured at 4 Kelvin and 10 Tesla fields. And we can kind of measure, when we run current through this really thin piece of material, we can actually measure where does the current kind of scatter, where does the current, where in these parts up there. Again, using this force microscope technique, essentially, we developed. So, for example, we've seen the top image there, where you can kind of see these filaments there. That is really exciting because it actually tells you something about defects in materials and where does something like resistance come from. Resistance is kind of useful, or in some cases, or a drag. Essentially, it's a loss mechanism. So most recently, the last 2 or 3 years, we've actually combined atomic force microscopy with ultra-fast optics. So we wanted to understand, how to separate charges, the role of defects, essentially to improve the efficiency and lifetime of solar cells and batteries. And so then you have to actually measure these single electrons that I showed you in the previous picture, time resolve. And typical time scales for these phenomena is really, 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 really fast, about 100 femtoseconds. That's the distance light travels. 100 femtoseconds, light travels about 30 centimeters. So it's really fast. So what we need to do again is we buy a bunch of really fast lasers, we incorporate them into our own system, we kind of figure out how to get the light into the ultra vacuum system. This is the inside view of the thing that you saw Sarah Burke looking into a few slides ago. We have to invent and then build and debug and make work little motors that can steer the beam onto the sample in this ultra high vacuum system. And in the end, you discover a whole bunch of really interesting new phenomena. So, for example, here is again, a 1 atomic layer thick or thin piece of material, molybdenum diselenide, and you can kind of see its photo response here, measured by force. So that's this blue part. You can see that somehow within 100 femtoseconds, it changes dramatically compared to the neighboring material, which is silicon oxide, essentially glass, where you see no photo response. And so from stuff like this, you can actually figure out why molybdenum diselenide is such an efficient photodetector. So with that, atomic force microscopy essentially is an endless frontier, because at the nanometer length scale, and what you see on this image are actually individual atoms of silicon, the stuff that your cell phone is made for, kind of start understanding how things work at the atomic scale, how their structure relates to their properties, and now we can actually improve on a whole bunch of science and technology relevant to society. And with that, I'm looking forward to having an interesting discussion with you guys.
Okay, fantastic. Thanks very much, Peter. Our next speaker will be Tomislav Friščić from the Department of Chemistry.
Tomislav, you should be able.
Woohoo! Okay, this seems to be working. Hold on. Is there any way I can turn my camera here?
No, you don't see yourself.
For some reason, my camera doesn't want to start. Okay, so let's see how we can get this one going. Sorry, guys, I seem to have a bit of a problem here with the keyboard. Anyway, let's do this. Hi, I hope you're all having a really good Friday afternoon. It's pretty sunny here in Montreal. I don't know where you folks are from, but I hope you're enjoying the day. So I came here to maybe spoil the day a little bit by telling you about some research we're doing here at McGill Chemistry. And first, what I want to do is actually show you the picture of my research group, which is a bunch of really enthusiastic postdocs, undergraduate, masters, PhD students. They're just a fantastic bunch. And as you can see, most of the time over the summer, they spend it having a good time, except obviously now in the coronavirus. I hope at some point they're going to let us go out and we'll be able to get together again and have a nice barbecue. Anyway, these are the people who do all the work. And what I like to talk about is chemistry from a very, very fundamental perspective. So I like to ask very basic questions, such as what is chemistry? And that's something that if you have taken undergraduate chemistry, or if you've taken high school chemistry, you probably know the answer for, and it's probably going to be something along the lines of reactant A and reactant B come together through some kind of a transformation indicated by this yellow arrow, which we call a chemical reaction, and they give us the product C. Now, this is a perfectly fine representation of a chemical reaction. You can find it in a number of textbooks, number of research papers. This is how we think about chemistry. This is how we teach chemistry. And I have very little problem with it, except one fact. It's actually incorrect. Because chemistry is not simply taking A and B as some sort of starting material solids and bring them together. We all know that actual practical chemistry, real-life chemistry, requires us first to dissolve this material, this reactant A and B, in a solvent. And once these A and B are dissolved in a liquid phase, in a dilute solution, then we can mix those and obtain a chemical reaction, not by mixing A and B, but solutions of A and B in a solvent. And then your product C also forms in a solvent, from which it's eventually isolated, extracted, purified, and so on. Now, there is absolutely no problem with that in the context of small-scale laboratory research. But on a large scale, chemical manufacturing, the way we make stuff globally, it's actually a problem. Because most of these solvents are organic liquids. They're not terribly friendly. Most of them are toxic, volatile, flammable. Some of them are actually greenhouse gases. So they can actually be pretty nasty. And imagine what? The scale of global chemical operation produces around 5 billion liters of waste solvent every year. So this is a vast amount of liquid waste that is actually being created and turned into chemical waste. So what my group is asking is, why do we need to use all this solvent? Is there actually an inherent need to use solvents in chemistry, or is there maybe another route? And it turns out there is another route, and it's been known for a very long time. On this picture, you can see some of our, presumably predecessors using rocks and stones and all sorts of gadgets to turn things into different things. Chemical manufacturing, materials manufacturing, medicinal sciences have relied on the use of solvent-free mechanical ways to make new materials for thousands of years. And this is actually what my group is doing. This is called mechanochemistry, chemical reactions induced by mechanical input, mechanical energy. And here is a nice little video of what we do. This is a picture of something that we call a milling jar, which is essentially a small plastic vessel, in this case made from transparent plastic. It contains a ball. This is why it's called ball milling, which is made of tough ceramic like zirconia. So if you actually, like me, happen to have a crown on your tooth, a white crown made of very stable ceramic, that's zirconia. So it's a very hard, very inert material. And besides that, in this jar, you will have these white and dark powders. What we do now is we take this whole assembly and we place it onto a shaker, an electronically controlled shaker, and we move it left and right. Now there is a movie, I don't know if you can see this, we are actually shaking this sample at a frequency of 30 hertz, which means that the sample goes back and forth 30 times a second. Now this is actually not anything violent, but it makes for a great video. And this is essentially how we do chemistry. This is a different way of doing chemistry, where we perform a transformation, in this case of a salt of a metal called europium to a metal organic product, rapidly by grinding. And even better, this movie has actually been recorded on an iPhone by a visiting undergraduate student from the University of Cardiff in the UK. And all he needed was actually a fisheye lens and some kind of app that slows down things, I think, 4 times. But what you can see from this, that even with such a slow motion video, the mechanochemical, solvent-free chemical reaction takes place very quickly. Okay, so I want to just very briefly tell you about what this is useful for. And I want to focus on gold. Gold is an amazing element. Everybody loves gold. Well, it's a fantastic piece of jewelry, but also because it is a fantastic thermal and electrical conductor, it's very stable. It's also central to most of our industries. All of electronics, computer gadgets, cell phones, none of that would really be as functional as it is if it was not for gold. So, but enough about using gold. How do we actually get gold? Well, gold is very stable, which is really a virtue, but it's also a huge curse, because you know that in order to do chemistry, as I told you, you need to dissolve everything. And there is only one way that you can dissolve gold. That's using something that's called royal water or aqua regia. It's a mixture of two very nasty substances, concentrated nitric acid, concentrated hydrochloric acid. Extremely nasty stuff, but it's very powerful and it can dissolve gold. We use it not only in the lab, we use it in industry to extract not only metals like gold, but also platinum, palladium, the sort of things that you use for clean catalysts in cars. And this leads to essentially creation of vast rivers and lakes of acidic, toxic wastewater, for example, in mining. So what can we do about this? Well, we actually want to help. So what we are thinking about is, if we don't need to dissolve gold, so if we can perform a mechanochemical process, maybe we can actually help clean up some of these industries. And here is a project developed by our current student, Louis Daw, who milled gold in the presence of insoluble oxidants instead of strong acids. And he converted gold here, gold and palladium, by milling it in 4 very colorful powders, starting from the metals themselves. And the cool thing about these powders is they are soluble in water. So we have a way to directly convert gold metal, palladium metal, platinum metal into soluble materials that can be directly used in processing, in extraction of elements, without having to use strong acids and creating toxic waste. And this is not really where it ends, but I really need to stop. What our group is really hoping to do is create something that we call Chemistry 2.0, an alternative, cleaner, safer way to do chemistry entirely or mostly without solvent. And we are working with industries, not only in mining, we are working with industries in cosmetics, in dye manufacturing, in plastics recycling, and also in pharmaceutical manufacturing. And with this, I would like to say, thank you for your time.
Fantastic. Thank you very much, Professor Friščić, for that very interesting talk. We'll now move next to our final speaker before the breakout rooms, Professor Virginie Millien, who's from the Redpath Museum.
All right, thank you. I think I'm gonna have to find my presentation here. Okay. So I'm going to talk about, it's going way too fast, but I was going to tell you a mouse tale in warming world. As you know, you're familiar with this type of curve. What it's showing you is the temperature anomaly. So you take an average over time, and then you look how it differs. And so everything above 0 is an anomaly. So it's warmer than the historical average, if you want. So what you see is that since the 1980 on that curve, it's pretty clear that it's increasing super fast. We're getting to 1 degree C above the normal. So this is what nature ecosystems and are dealing with, have been dealing with. And so my lab is interested in knowing, well, whether species can cope with that and how do they do it. So I can't change slide. What we've been doing, this is very annoying.
Jenny, do you want me to flip through the slides and just let me know when to flip? Is that easier?
Maybe.
Okay.
Oh, it's back to.
Just let me know when to go to the next one.
maybe I got too excited on my keyboard and it kept shifting way too fast, but there's a lag. So on the next slide, what you're going to see is one of my students ran some different models for all species of mammals in Quebec. I don't know if you guys are aware, but there's nearly 60 species that are resident in Quebec today. And each dot on that graph represents one single species. And what we've been doing is looking at where they are now. So that's all the little current maps of North America. Quebec is here in the north. And where they're going to be in 30 years by 2050 with such climate warming. And we observe a few type of responses that I put in different colors. So I'm highlighting a few. This is interesting. I promise you I can spell better than that. I don't know what happened there, but that species here is losing a lot of its area in the future. It's going to lose over 40% of its whole range. So range contraction, its a habitat is not suitable anymore. So what's happening here at the southern range of its distribution is local extinction. And you can imagine that in time, it's going to get worse and worse. And eventually that species is a good candidate for just becoming extinct altogether. Other species are moving up north. And this is shown on that axis, the y-axis here, is the shift in latitude. So everything that's positive, and they're all, most of them are positive, is moving up north. So this is the case for this chipmunk, for example, where its distribution is simply shifted a few degree latitude up north. So it's tracking the climate, really. It's like, well, you know what, this is getting too hot here, I'm going to move north. And then there's another type of response here. It's range expansion. So not only is this shrew, which by the way is as big as a snail, it's the tiniest mammal that we have in Quebec. So it's super cute despite being a shrew. And it's expanding its range. So this one is a winner under climate change, right? Under climate warming. It's going to be able to occupy a wider range of habitats. But overall, so what you see from that is that there's a couple of responses. Number one is extinction. Number two is shift in distribution, whether it's expanding or not, but shift in distribution. There's a third response that I'm going to talk about in a minute. But from that graph, what you see is that all these Quebec mammals, most of them, a vast majority of them, are going to shift up north. So we're going to see an invasion from US mammals, species from the US invading Quebec. The net effect, in the short term, I'm talking a couple of decades, is we call it the Quebec biodiversity paradox, because the net effect is going to be an increase in the number of species of mammals. And that's a paradox because you all know that we're currently in the midst of a mass extinction, the sixth extinction. Now, don't get too excited. We're not going to have more mice and squirrels around for a long time. This is only temporary. Obviously, that's going to stabilize and go back to the normal, if you want, normal number of species in a bit more time. But still, that's very exciting time for us scientists looking at all this happening in front of our eyes and live. So these are models, but I want to show you, okay, let's go to the next one. I want to show you what we do more locally in the field. This is happening at the Galt Nature Reserve. It's a field station of McGill University, the Faculty of Science, actually, just south of Montreal. I was there yesterday, and it was a lot of fun. And what we've been doing pretty much for over 10 years now, I've been conducting inventories of small mammals. So small mammals, think rodents, mice, shrews, And we've been capturing them, counting how many they are, where they occur, and so on. And then comparing to what we knew from the past, from 50 years ago. So that pie graph here on the left shows you all the different species. Each color is one species that were occurring at the Gault Nature Reserve around the 1950s, 1960s. So there were 13 species at that time. Some abundant one and some not so common. And the most abundant is this orange one, the deer mouse. I went back, and what we find today is a complete different picture. The pie diagram, same color for the same species, now has lost a number of species. There's only 8 species left. So there's less species. There's been a decrease in the number of species and a complete change in the composition and who is the most abundant or not. Nowadays, it's not that orange mouse, the deer mouse, that's the most abundant. It's the red one, the white-footed mouse. And here they are collaborating very happily to our field sampling and staying in their traps nicely. So we find a lot of these now at Gault. I wanted to look at what's happening with the species. So we know it's shifting its range, it's invading Quebec. It's traditionally in the past, it was found in the US, now it's crossed the border and becoming more and more abundant in Quebec. And I'm going to talk now about the third response. Remember, number 1 was extinction, number 2, range shift, as illustrated here with the white-footed mouse. The third response is local adaptation. Yeah. And local adaptation is here. So, and I'd call it evolution in action. What we did with these mice, we measured landmark positions of points on their skull. This is a mouse skull with the teeth here that you see, the nose, the rostrum, and the brain case. So we record the position of a number of points on the skull. And we looked how it changed in time. So we took museum specimens from the 1950s, called them historical, and then our specimens that we process, prepare the skulls and measure them. So that's today. And then we do all sort of statistics and data mixing and computing on that and produce what we call a shape axis. So it's summarizing the shape of the skull. That's the first axis, but never mind the details. What you want to remember from that is that between 1959 and 2014, the 2 mice that we were looking at Gault have changed the shape of their skull. I'm talking about a bone structure that has evolved in less than 50 years. So that's pretty amazing. And we think in my lab that this is in response to the change in climate and associated change in habitat, of course, and the type of vegetation that can occur on the mountain. And so here we go. We've now documented the 3 types of responses for climate, of mammals to climate warming. And it's every time, I can tell you, every time we look at a new question and a new species at a new place, we find evidence of that. We find evidence of the effect of climate warming on all aspects of mammal biology. And this is cool, but this is also worrying because with that comes some unexpected consequences. The white-footed mouse, the one that is evolving and apparently getting pretty happy settling in Quebec and taking over, becoming really abundant, is also bringing with it, is the preferred host for these nasty little guys here. This is a black-legged tick. If you guys are from the US, you're most likely very familiar with this because they carry Lyme disease, and these ticks can bite humans as well as mice. They prefer mice, but they'll go for humans if they can. And that's how you get Lyme disease. And so it's becoming a concern in Quebec. It was not a decade ago. Lyme disease was not occurring in Quebec, and now it's endemic to southern Quebec, meaning there's an actual risk of getting it. And all of this is happening because the mouse has shifted its range and has become established in Quebec, so it can now spread the disease. This is one example, Lyme disease, but there's many other examples of zoonotic disease that come from animals that are moving around with their host, and climate warming is really messing up with all these disease system and helping spreading that. On that, I thank you for being here with us today, and I'll be happy to answer questions in these breakout rooms in a minute. Thanks.